Hello and welcome to another Prof episode. For this episode, we'll be talking about the researcher Dr. Melanie Kalischuk. She's a specialist at the University of Guelph who studies the genetic changes in plant pathogen populations and the gene pool of different plants. She also teaches the funnel project courses where she matches professors with students for small research projects that can be done 1 year, or more specifically 24 weeks. In this episode, we'll be covering a paper by her regarding the genetic changes in late blight. Let's get started. Late blight is the most devastating disease in potato. It's caused by the oomycete Phytophthora infestans. Oomycetes are a family of organisms that exist in the intermediary between protozoa and fungi. This species infects both potatoes, tomatoes, and other solanaceae crops. It reproduces both clonally as well as through sexual means. There's been a gradual increase in genetic diversity within Canada and the United States of this pathogen. These strains have different levels of virulence as well as resistance to fungicides. This resurgence has been attributed to the spread of clonally propagated spores that have been moving long distance. Disease losses within North America are caused by two specific strains of late blight, A1 and A2. Both A1 and A2 were introduced from Mexico in the 1980s. The current migration elements that are happening in terms of clonal specimens has led to the replacement of the A1 variants with more aggressive A2 variant, and the mingling between both populations has created new genetic combinations. Sexually reproduced spores, unlike the air spores, have thick cell walls that are capable of surviving in the most extreme temperature, moisture environments found in Canada and the United States. This means that the management of this disease will become more complicated as time goes on, due to new resistant strains to different fungicides showing up, and new recombinations possibly increasing the amount of virulence and aggressiveness within the pathogen itself. To figure out how many recombination events are happening within Canada, late blight samples were obtained from potato, tomato, and other host crops. These samples of the disease were obtained from both field and greenhouse environments. These samples were then cultured out on top of tissue culture medium to produce a large population within a petri dish. These populations were then subject to genetic analysis to see how much variation was in between each sample was found and how much recombination was found. 138 isolates were collected from potatoes and tomatoes in 2011 from 8 provinces all across Canada. These include British Columbia, Alberta, Saskatchewan, Manitoba, Ontario, Quebec, New Brunswick, and Prince Edward Island. Most of these lines seem to belong to a small number of different strains, 2 of which belong to the United States. The strain labeled US 23 has been found in BC, Manitoba, New Brunswick, PEI, and Saskatchewan. The United States strain US 11 was found in Ontario. Later discoveries found that Alberta, Manitoba, and New Brunswick and Quebec had a new strain from the United States called US 24, and there's migrations of US 22 genotypes found in rare spots in Ontario. The currently existing strains within Canada seem to be becoming more resistant to common fungicides. 2 of the common genotypes, US 8 and US 11, are now mexalaxyl insensitive, while the new strains from the United States, such as US 24, are still sensitive to this fungicide. This is pretty good on a genetic front because US 24 is replacing and not competing US 8 within New Brunswick. Only the A1 strains were found within Western Canada, specifically British Columbia, Alberta, Saskatchewan, and Manitoba. Within Eastern Canada, which includes Ontario, Quebec, New Brunswick, and PEI, the late blight strains are either A1 or A2 mating types. In spite of both populations being able to crossbreed, no examples of crossbreeding have been found within these regions that seem to be atypical. However, genetic diversity has been increasing exponentially within Ontario, where both mating types are incredibly close. Most strains seem to be derived from the United States strain US 22. 10 of the 17 lines to be derived from that. Further genetic analysis also found some recombination events in various parts of the pathogen gene pool. specifically segments 1, 5, 14, 18, 18, 20, 21, 24, and 25. New strains have also been found designated CA9 and CA10 and C11. These strains seem to be combinations of the US22 and US11 genotypes. Based on this research, it is likely that most of the genetic diversity that's appearing within Canada regarding late blight is a result of the natural movement of strains from the United States right into Canada. This is further being exacerbated by the creation of new lineages made from the mixing of both the old and new varieties within Canada. Some of the older strains are becoming more resistant, and the newer strains are more aggressive. Those derived from both lineages could be the worst possible combination for us. As of right now, the two most common varieties, A1 and A2, are found primarily in Ontario, and exist in close proximity to one another. As a consequence, although there's very little recombination events through crossbreeding between both lineages, there are some, and some is enough. Dr. Kalischuk concludes that late blight epidemiology should be improved, as should the tracking of this pathogen, and its management should be improved, and that will. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.